So you do have an exam scheduled for this week. I'm going to push that back a little bit towards the end of the week. I'll give you till Monday of next week to finish. So it'll be exam 4. I'm going to have chapters 25, 26, 27, and 28. So I'll be lecturing on 28 tomorrow, finishing up 27 today. Okay. I've received a few emails about the project, which has been scheduled since the beginning of the semester. So I'm a little disappointed that a few individuals don't know what's going on still. So we do have a paper presentation and or a paper and a presentation that's due. Today was supposed to be the first day of presentation recordings or presentations to myself. So I posted on week 11. We should have had a paper topic turned in by October. But you knew about this a long time before. And if you don't remember, it was the recordings that I showed you, IFL Science, where you would choose a topic of your choice on any relevant science topic to date. And it didn't matter if it was in biology or another type of science. I just wanted you to write a paper on a relevant science topic and then have a little short 10-minute presentation that's due. Okay. So the oral presentation, it was going to be about 15% of your grade. I was going to provide an extra 5% of your grade, or 5 points were going to be extra credit for those of you who did really well. And I gave 5 points of extra credit, but over half the class hasn't even signed up yet. So please make sure you go in and sign up for that. And I believe I've shown this multiple times on the recordings. So I'll show it one more time how to sign up. So there's presentation schedule. So it's the schedule for presentations right here in week 13. So just go 2 weeks back and it gives you, you click on the link and it gives you this Excel sheet to access, and you have multiple days to sign up. Okay. So the majority of the students have already taken up the 9th and the 10th. There's still 7th, 8th, 2nd, 3rd, 1st, 30th, and then there's tomorrow and today, but no one has signed up for any of these time slots. Okay. If this time slot does not work out for you, we can try to work in a time that we can get you in on a different day. That being said, if you don't complete the presentation, you're going to lose about 20% of your paper grade, which is 100 points. It's worth the same as an exam. So if you miss that paper, you're losing a fifth of your points on your paper already. So you're already starting off with a C, okay, without having me grade your paper yet, if that makes sense. Okay, so this is 20%. It's a chunk of your grade, okay? So please make sure you schedule a presentation. Please make sure you are prepared on that day. Again, it's very informal. You're gonna pop up on that time slot for that week that you're, or the day and time that you're in. You're gonna just come in on Zoom for 10 minutes and you're gonna give your presentation. And you're just gonna talk about your project. So it'll be a summary of the paper that you've written that's gonna be due at the end of the semester, right? I'm going to ask you a couple questions about your assignment or your presentation, and you'll be graded on that as well. And then you're done. You chime off. Okay. You may have a PowerPoint presentation. You may not. You can, orally talk and just give a presentation rather than having any type of PowerPoint or graphic. It's really up to you. Okay. I just offered to have, if you wanted to have a couple, like 5 slides that you can go through in 10 minutes, that'd be more than enough to be helpful. Okay. So go ahead and look at the assignment and make sure you get that done. All right. So yeah, the fact that today's the first day of presentations, I wanted to see if anyone would get it done early. It looks like no. So people should no one has to give the present, essentially, in reality, no one has to give presentations on today for the time slots to be filled. Um, if everyone decided to sign up, we would be full from the 30th all the way to the 10th. But I opted to give early presentation times for this week just because, um, I'm going to push back the exam till late, early next week. So yeah, if anyone has any questions, please let me know. But the assignments there, I re-uploaded the assignment just because I had a couple of students um, email me asking for the information on the assignment, and I went ahead and put it again in week 15 for this week. Okay, so presentation rubric. Okay, paper and presentation rubric. Right, this is all the information you need. The sign up is in week 13, schedule for presentation. So you're going to go ahead and sign yourself up, right? It's going to take you to this page here. You can literally write in your name. So, right, Eric, right, A, that's me. But no one's signing up today, so we're not having any presentations. All right, let me finish chapter 27. I think we were here on sensory input. I think we were here. All right. So sensory input, okay. All right, in order to have a reaction or have the central nervous system react to the peripheral nervous system, we have a few different stimuli that need to occur. Okay, so here we have a taste bud that's reacting to an ice cream, right? And these are going to be taste sensors. These are going to be something that's going to receive a chemical signal that's going to induce the taste buds to give a neurological signal, right? And this is very similar to um, our pituitary gland and our hypothalamus, right? So we talk about how hormones play a key role in maintaining homeostasis within our body. Okay, so the hormones are our endocrine system are going to be providing some sort of signal that is not electrochemical, right? What we talked about the nerve firing. This is kind of a bridge between the two, right? So this turns a chemical stimuli, right? Sugar, which is chemical stimuli, into an electrical stimulus, right? So here we have that chemical stimulus, sugar, inducing a signal transduction pathway, allows for ion channels to react. Right, we talked about the firing a nerve, and then we now have acetylcholine or some sort of neurotransmitter uh, inducing an electrochemical chemical response. Okay, after the sugar was provided to that cell. Okay, after that sugar was given to that receptor. Okay, all right. So this is a sensory input. That sensory input is going to be that sugar chemical stimulating the nerve impulse, right? So you can lick an ice cream cone or something sweet, and you have that instantaneous, near instantaneous sensation of tasting sweet, right? And our brain understands that, right? And this is a chemical being turned into a neurological or an electrochemical, okay? And the electrical chemical are those ion channels, right? Inducing the firing of that neuron, okay? And again, remember the neurons, right? We talked about the firing of the neuron, um, is this electrical pathway, right? The turning on and off, the firing, right, of these axon. Okay, um, this is an action potential. We don't remember, right? The flipping or the inversion of these charges, allowing for these sodium, potassium ions to change. Okay, so one negative, one positive, boom, and they fire. It's like thinking of the domino effect. Okay, so the chemical stimuli would be the sugar, and then we would get the electrochemical response here, which is a firing of the neuron. Okay. So remember that. Okay, remember that as a firing or the receiving and sending of that signal. Okay, so there's a few different types of sensory receptors. We have pain receptors. So these are receptors that detect painful stimuli. Okay, we have thermoreceptors. So sensory receptors that detect heat or cold. Mechanoreceptors, a sensory receptor that detects physical changes in the environment associated with pressure, touch, stretch, motion, and sound. Electromagnetic receptor, a sensory receptor that detects energy of different wavelengths, such as magnetism and light. Okay, and these are just a few different type or 5 different types of receptors. But we have heat, light, pain, and cold. So thermo, um, uh, we have the electromagnetic detection here. And we also have pain. So cold, heat, light, electromagnetic, and then pain receptors. Okay. So these are going to be just under your epidermis. So in your dermal layer, you'll have your nerve endings and the innervation of those nerves. Okay. Um, this is CNS. This is not the CNS. It's going to be the peripheral. Okay. So this isn't your tissues. Remember, think of the CNS. I don't know why sometimes these are confusing. The central nervous system is your brain. It's going to be your brain and your spinal cord. Okay. Your peripheral nerve system is the networks of other nerves that are leading from your spine. Okay. Let me see. There's a good picture that I saw. But yeah, your there. Where's that picture at? The nerves. Oh, here you go. Yeah. So the central nervous system, okay, brain and spinal cord, the nerves that are distending out and innervating your tissues and your distal appendages, like your hand, your arms, and your legs, is your peripheral nervous system. Okay. So that kind of disregard that description here, nerve to CNS. Okay. It's a little inappropriate. All right. Um, but essentially we have the innervations of different receptors here on your skin. Okay. You have nerve endings in your hair follicle, right? And so we can feel the pain if you rip your hair out, pressure, and the amount of nerve endings on different parts of your skin are going to be either less or more, right? There's actually less receptors found on your back, whereas if you look on your hand or your thick skin on your hand, there is much more receptors, right? Just because we utilize our hands for different things, for touching or for special intricate jobs, right? Whereas the back, you really don't have that much use for having multiple pain receptors or different types of receptors, right? So there's different areas on the dermis or epidermis that'll have more or less innervation of the nerve endings than others, okay? Which makes sense, all right? So we have different types of receptors, electromagnetic receptors in the eye too. Okay, so we have a retina. This is a light sensitive layer in the eye made up of a photoreceptor cells and sensory neuron. Okay, we have a cornea, the transparent front portion of the white connective tissue that emits light into the vertebral eye. Iris, this is the color part of the vertebral eye. Muscles of the iris regulate the amount of light that enters farther into the eye. Okay, so low light situations, the iris can dilate, and um, large or very large amounts of light, your iris can contract and limit the amount of light that can get into your eye, right? So in the sun, you show your eyes, or if an officer were to check to see if you had been drinking, he or she's looking for the contraction of your iris, right? Um, and depending on if you're impaired or not, your iris can contract at different rates. Okay. Same thing if you were on some sort of narcotic. But if you ever close your eyes and open them right when you walk into a room, your iris will contract. Right. The more light there is, the less light there is needed for the eye to actually see and sense. Therefore, the contraction of the iris will occur. So it'll close off. Okay. You have a pupil. This is the opening in the iris that emits light into the interior of the vertebral eye. Muscles in the iris regulate its size. So lens, the disc-like structure in an eye that focuses light rays onto the retina. Okay. Path of the light to the retina. Once the light passes through the eye, it is focused on photoreceptor cells in the retina, right? So here we have, again, the iris, which is going to be the contraction or opening. So this is that the black part of your eye, remember, that gets larger or smaller. That black portion of your eye is what allows light to pass through, um, so it can hit your retinas. Okay, so your retina here contains the cones, and other types of sensory cells. Okay, so this is what will allow your cells to receive the light. Okay, and this is what you're able to see with, right? Um, Remember, your sclera is the white part of the eye. Excuse me. Your cornea and your disc right here, your lens. You can get corneal transplants, right? And it's going to be this uh, sheathing right here. Okay, this cover, this nice little disc. Sometimes this lens can be replaced as well, especially if an individual has glaucoma or some sort of infection. You can have corneal transplants. You can have the disc transplant as well. Um, this is actually not as difficult to do as if you were to have a liver transplant. And this is because actually the eye is void of the body's immune system in a way. Okay, so actually immune cells cannot get into the deep tissues in the eye. Okay, um, which is very important to think about because this would mean that if there is not a lot of immune innervation in the eye, which means that your body really doesn't receive any check systems in the eye, you can receive corneal transplants or different types of tissue transplants without the risk of having your body reject the organ, okay, that you're going to be placing, or the tissue that you're going to be placing into the eye, which means these surgeries are relatively safe, right? If someone were to receive a heart or a liver or a kidney, they would need to be placed on immune suppressive drugs for the rest of their life, right? Because if you give someone a different organ or a different or a new tissue from a different person, your body will mount an immune response to it. Okay, whereas in the eye, since the eye is void of innervation of the immune system, you lack the need to really treat and provide immune suppressive drugs for that individual, right? Which is really important, which means it's relatively safe if that tissue is taken out by that individual, if it actually works, right? Whereas if someone receives a liver, again, they're going to be immune compromised forever now, okay? Because they are taking medications to stop the body from reacting to the kidney or the liver or whatever organ system, new organ that they place into their body, okay? Whereas here, we really don't have to worry about it, again, because we don't have it, we lack the innervation of the immune system in the eye. Okay, so kind of keep that in mind. We do have something called a blind spot, which is because the way that our eyes developed evolutionarily speaking, we have the nerve um, grew into our doesn't sound weird. The nerve grew into our heads and then developed the eye. Whereas the eye was not a photoreceptor previously, like in squid or mollusks. And so squid and mollusks actually don't have a blind spot, right? So what happened with humans is the nerve reached out, grew into the cavity, which then became our eye socket. Whereas in mollusks and squid, their eye was their eye was once a photoreceptor or photoreceptive light cell patch that eventually turned into an eye, but the nerve grew away from that receptor, or those receptors. I guess I should show you. I feel like it's doing me explaining is doing it just a disservice. Okay. Let me see. So squid Squid or octopus eyes actually do not have um, the blind spot, right? So here we have a human eye, and this is showing the photoreceptors here. Okay, um, so these are all the photoreceptors here, but you see how the nerve had a puncture through into the human eye, right? So the nerve grew out and then in, and then the eye developed. Okay, so the nerve had to reach out and extend almost. Whereas in an octopus eye or a squid eye, these cells were already present there in a primitive form. Okay, so a primitive eye was already there, and then the nerve grew away from that eye. Okay, so this was already there, then the nerve grew away, and that's an octopus, right? So they lack the blind spot, whereas we have a blind spot. Okay, right? We have a blind spot here. An octopus eye, you see, you have the eye develop, and then the nerve grew away. Human eye, a nerve had a puncture into the cranial cavity or into the common ancestor that had some sort of a head, and then the development of the eye persisted that way. So we have two very distant lineages of development. Okay, and the eye points to that because squid eyes or octopus eyes are very sensitive, or even they're much more sensitive than our eyes, right? But they also lack the blind spot. Okay, so show right. So octopus eye. See again, you don't have the blind spot here. Okay, so very different evolutionarily speaking, in development. Okay, right. Where we're humans, we do have a blind spot. Okay. Oops, wrong PowerPoint. Okay. All right. So how the lens of the eye focus light. When viewing a nearby object, near vision, the lens becomes thicker and rounder and focuses the image on the retina. When viewing a distant object, distant vision, the lens becomes flattened and the image is again focused in the retina. Okay. This is talking about nearsighted and short or nearsighted or farsighted individuals that need glasses, right? There's something called astigmatism as well, which means that the eye is misshapen. Therefore, it doesn't allow for the focal point of the eye to be accurate, right? So one eye is misshapen from the other. Therefore, they do not have the same focal point. So this is when you see some individuals will have glasses and then they'll have one glass prescription, one lens prescription is different than the other. Okay. And again, it has to do with the shape of the eye and the ability for your eye to focus light. Okay. All right. Oops. All right. So the retina contains a couple of types of cells that can pick up different types of light. Okay. So here we have a rod. So your eye contains rods, which allow you to see dim light, and cones, which allow you to receive color in bright light conditions. Right. So again, rods are able to see dim light. Cones are able to see distinct colors within brighter light. Okay. So we do have synaptic terminals, right, that receive the sensory input. Okay, so these cells here, this rod will see the light, and same with this cone area. The sensory input is received and then it will be sent down to the synaptic terminal. So there is going to be some sort of firing or activation of a response. Okay. So the membranous disc contain visual pigments. These visual pigments can become excited with light. Okay. So think of photosynthesis and the activation of the vibration that's caused by light. This will be the same stimuli that you'll see there. Okay. All right. So here you'll see again the retina, which contains the photoreceptive cells. So you have the neurons, the photoreceptors, and the cone, right? Cone or rod, right? So remember, the rods receive information from dim light. Cones receive information from brighter light in terms of color and visualization of color. Okay, So again, the light can focus down through into these receptors, bounces off the back of the wall, the eye, and then you pick up the light, and then or you actually have a stimuli produced in the cones or the rods that can then trigger the nerve. Okay, so the optic nerve goes directly into the brain. This can directly pick up any type of changes in those cones and the rods, therefore provide a picture or a visual image that your brain can interpret. Okay, um, and again, this is electrochemical receptors, right? They're picking up visual light. Okay, so between, I believe it's 280 to 300 nanometers to about 900 nanometers of the spectrum, the electromagnetic spectrum. These cells can pick up uh, the light. Okay, so visual light. Okay, so hearing. So you have an outer ear, one of three main regions of the ear in humans and some other animals. The outer ear is made up of the auditory canal and fleshy structure that we commonly call the ear. Eardrum is a sheet of connective tissue separating the outer ear from the middle ear. The eardrum vibrates when stimulated by sound waves and passes the vibrations to the middle ear. Middle ear, one of the three main regions of the human ear, a chamber containing three small bones that convey vibrations from the eardrum to the inner ear. We'll see this right now. The inner ear, one of the three main regions of the human ear, includes the cochlea, which contains a hearing organ. Okay, the cochlea, a coiled tube in the inner ear that contains the hearing organ. And this will look almost like a snail. And you'll see that right now. Okay, so here we have the cochlea. Again, you'll have um, different structures that allow for hearing in here. We have the eardrum, which can rupture, and then we have the anvil, stirrup, and the hammer. Okay, and these are all going to be bones that are actually inside of the ear that allow for vibration to occur and allow for increased, or allow for the transfer of that sound to the cochlea. Okay, here we have another organ again that's going to allow for the islets that are going to allow for us to determine whether or not we're balanced appropriately. Okay, there's going to be a little mineral rock inside of this organ here that allows for us to balance properly. It gives us orientation in the world. This can be messed up and cause it'll cause vertigo. Okay, so some people have they what the doctors typically say they need their crystals set, and the crystals are actually housed in this little bony structure here. Okay, and that gives you orientation, right? That crystal will be sensitive to gravity in terms of it'll allow you to be able to stand up straight. You know that you're standing up straight. Okay. Um, you can rupture this eardrum here and damage your hearing. Rupturing the eardrum either can be from a loud noise or you can rupture it from a drop or change in pressure as well. So sometimes individuals that do like high skydiving or they'll go diving in the water can actually rupture, push through this membrane, or the change in pressure um, from jumping out of a pressurized airplane out. The drop in pressure will actually suck the membrane out and cause a rupture that way as well. Okay, and car accidents and severe trauma can do this as well. So getting like hit really hard, causing that severe change can cause issues as well. Okay. So here the cochlea is a coiled tube containing sensory hairs and flexible membranes. Sounds cause vibrations of the basilar membrane, forcing hair-like projections on hair cells, mechanoreceptors to bend. Hair cells convert the stimuli to receptor potentials that are communicated through the auditory nerve as action potentials to the brain. Again, this is stimulating that mechanoreceptor, these mechanoreceptors here are going to be stimulated and send the action potentials out towards the brain and picking that up. The brain converts that into something that's tangible for us to understand. Okay, so here we have the bone of the cochlea and we have the individual hairs that are present. Right, these cells then have these hairs that vibrate as the membrane vibrates. Okay, so remember these the cochlea is connected to these different bones that will vibrate as sound or stimuli comes into the inner ear and causes this motion or movement of these bones. Okay, and that's transferred to the cochlea and these hairs, which can then will move depending on the amount of stimuli that's there. Okay, and you can damage these cells. You can provide too much stimuli and essentially break these hairs or damage the overlying membrane to the point where you cannot hear anymore. Okay, so you can have damaged hearing. All right, locomotor systems, so motor output essentially. So this is a reaction to the stimuli, okay? So the skeletal system. The skeletal system, the organ system that provides body support, protects body organs such as the brain, heart, and lungs, and anchors the muscles. Endoskeleton is a hard interior skeleton located within the soft tissues of an animal, found in all vertebrates and a few invertebrates, okay? So the skeletal system is in us is considered an endoskeleton. So think of starfish. Starfish are those echinoderms are going to be those that bridge between humans and primitive non-skeletal organisms. Okay. All right, and there's going to be a few bones here. We have femur, tibia, fibula, radius ulna, humerus, clavicle, scapula. Right. There's going to be a couple of bones I might ask you about, and I think they're going to be the main ones. So I do want you to know humerus, radius, ulna. So humerus is your arm bone, radius, ulna. Okay. Femur, tibia, fibula. I'm just going to give you an anatomical structure like this. Okay, and I'm going to ask you to identify. Okay, pelvic girdle. But you should be able to identify a couple of the bones. Okay, so just the major ones. Okay. Yeah. So there's a couple of different joints as well. We have ball and socket joints. This provides the most movement, okay, than a hinge or a pivot joint. So a ball and socket joint, right? Think of your shoulder, provides almost near 365-degree rotation of your arm, okay? These joints are the most sensitive and the most prone to injury, which makes sense. There's a lot of people with shoulder problems, right? But think of why. I mean, there's so much movement in that joint. There's so much innervation of muscles, of tendons and ligaments in those joints that they're more prone to damage and injury there. Okay. So it's a quite common shoulders are quite common to injure. Okay, we have a hinge joint, so think of your elbow movement. So this allows for single direction extension and flexion. Okay, so it's like an open and close kind of joint, right? Opens and closes, opens and closes. There really should be no movement other than extension and flexion. Okay, but you can have supination as well, right? So movement from movement of your hand up and down. So like if it can face down or it can face up, depending on in terms of your palm. Okay, that's mainly from the ulna and the radius having different articulation points in your wrist as well. Okay, a pivot. Think of your elbow with a partial rotation. So this is where you have your ulna and your radius that are able to turn, right? Supination and supine, right? Supine or prone, or supine or down, depending on this movement or depending on the joint. This is also similar to your knee, right? The knee is technically a hinge joint, but it does have um, uh, lateral or turning rotation as well, where you really shouldn't turn that joint too much under weight bearing or weight pressure movements because you're prone to damage. But again, those are more of those simple joints. You can see the pivot as well. Actually a joint that's good to talk about in terms of hinge is really the knee. You really don't want it to turn too much. There really shouldn't be too much movement there. All right, structure of an arm bone. So here we have cartilage on the edges of the bone. Again, this is going to be for articulation or those points that touch other bone. Okay. So if you see here the humerus, in the socket, you do have the articulation to the scapula as well as um, Okay, the scapula, humerus, and what is this here? Oh, I guess it is just the scapula. I thought it articulates with the it's just scapula. So here we have the articulation of the scapula. This is going to have a lot of movement. Ulna and the radius also will have articulation here at this cartilage point. So cartilage, essentially think of bone on bone contact. If there's bone touching bone, cartilage is typically present there, right? This is where you have individuals with issues in their knees. Let's say if someone was a runner, someone was always on their feet constantly and constantly weight bearing, they can damage your cartilage, right? There's not a lot of cartilage that might be left in some on a jogger's knees that has been jogging for 40 years, right? The cartilage can get worn out, right? But this is bone and bone articulation. This is bone on bone touching. This cartilage provides a level of lubrication. It allows for sliding, easy movements. Okay, so it's important that the cartilage is there. Um, you have spongy bone, so it contains red bone marrow as well. And think of this as areas of the bone that provide or make red blood cells. They also make platelets. These are the areas as well that house activated B cells, so plasma cells. And you have certain special cells like megakaryocytes that produce they produce your platelets, right? So these are almost think of them like totipotent or pluripotent cells. So think of it almost as stem cells that you'll house in the blood in the red bone marrow, right? You have the cells that'll make the red cells or the reticulocytes or erythrocytes, okay? Um, and then you'll have the cells that'll make the platelets. You have the cells that'll make the white blood cells, okay? So there are specialized cells in the red marrow, okay? You have yellow marrow, which is essentially fat. So if you've ever consumed bone marrow, and they serve you the bone and the chunk, right, where it's compact bone, and then you have that jelly-like stuff in the middle, that's essentially fat. Okay, the yellow bone marrow. Okay, um, you do have circulatory innervation of the bone. Okay. So this bone heals very quickly, right? There's a lot of blood that flows through compact bone as well as spongy bone. Okay. So this blood flow in the different osteocytes of the osteon. So you'll see these circular um, areas, the Haversian canals that have the innervation of these red of these of the circulatory system, so all this blood flowing through the bone allows for quick healing, right? Whereas if you look at the cartilage here, there's not a lot of blood innervation. There's not a lot of blood that can diffuse from these canals and then get to the cartilage. Okay, so it's important to note that cartilage, once it wears out, is very difficult to get back. Okay, and this is because you lack um, uh, blood flow to those areas, right? So like your back, even your cartilage in your back is very difficult to repair. The cartilage in your knees, the cartilage in your shoulder, the cartilage in your elbow, any of the cartilage anywhere is difficult to repair because again, you lack a lot of blood flow. People that get their cartilage pierced in their ears, that never really heals ever, and it's not that it's not healing or repairing itself. It's just that cartilage takes such a long time to heal. Your skin will heal over that hole that you made in your ear, but that cartilage will take a very very long time to repair. Okay, just because you lack the same similar amount of blood flow that you get from in your bone, right? So I've heard a lot of physicians say it's much more difficult to repair a sprain or a tear in your ligament or tendon than it is bone, right? Where you can just slap a cast on someone that broke their bone if it's not too bad of a break, and that bone is healed within a few months, completely healed in a few months. Okay, so bone is an amazing hard supportive tissue or supportive structure in the body that can heal very very quickly because of the amount of blood flow that bone receives. Okay, whereas cartilage does not. Okay. So skeletal diseases and injuries, we have arthritis, a skeletal disorder characterized by inflammation, inflamed joints, and deterioration of cartilage between bones. So we have just destruction or loss of the architecture of the bone or the cartilage. Okay, this is very common, right? Especially in older age, you can have arthritic joints again because of overuse and overworking. Again, cartilage takes a long time to heal. It's difficult to heal due to the lack of innervation of blood. Rheumatoid arthritis, this is an autoimmune disease in which the joints become highly inflamed. So autoimmune is referring to the body attacking itself. Think of auto or self-immune, self-immunity. So you're making immunity to yourself, right? Rheumatoid arthritis can be is a relatively common disease in the States. It's kind of a Western medicine disease, or a disease that's found a lot in the States. Again, and it's just your immune system is overreacting, and it's causing destruction or deterioration in joints. So sometimes you'll see balls or a fluid filling around the joints in severe amount of pain. Um, yeah, and again, it's your immune system is reacting to the joints in an inappropriate way, and it's attacking those joints. Osteoporosis, so is a skeletal disorder characterized by thinning, porous, and easily broken bones. Osteoporosis is common among women after menopause and is often related to low estrogen levels. Okay, so again, this postmenopausal women. Again, they lack the bone structure that or the development that typically occurs pre menopausal. Okay, and again, related to low estrogen levels means that the hormone is really dictating the amount of bone that is being laid down and being produced. Right. This is why they say it's important for women also to lift weights or have some sort of load bearing exercise or just move in general. Right. The more bone you develop early in life, the density of your bones, the better off you are. So it is good to have weight bearing exercises built into your cardio, whatever you're going to do on your weekly regimen, right? Especially for women. Okay. Um, not just men can lift and work out, right? It's also important for women, again, because the more bone density you pack on early on in life, typically the better off you are. Okay. The more developed musculature, your body is very resourceful in terms of dealing with certain stresses that you put on it. And if you start putting on stresses early on, you can develop thicker bone, more muscle, and that should help with osteoporosis later in life. Okay, the muscular system. So all the skeletal muscles in the body, skeletal muscle, striated muscle attached to the skeleton. We've just tested on that, so you should know what skeletal muscle is. The contraction of striated muscle produces voluntary movements of the body, which we should also know, right? We know that we can contract skeletal muscle, right? Biceps are triceps, right? Extension, biceps, flexion. Okay. Myofibril or tendon, fibrous connective tissue attaching to the muscle to the bone, whereas ligaments are going to be bone to bone. Okay. Myofibril, a contractile unit in the muscle cell made up of sarcomeres. Longitudinal bundles of myofibrils make up a muscle fiber. So sarcomere is a fundamental unit of muscle contraction composed of thin filaments and thick filaments. Thin filaments slide past thick filaments to shorten or contract the sarcomere. Okay. The contractile apparatus of skeletal muscle. So here we have the striated muscles, right? They have the very organized structure, very organized shape. And again, these are going to be voluntary, right? And here we have tendons, right? And ligaments would be bone to bone. Tendons, again, muscle to bone, and allow for movement. Okay. So muscles, it's important to note, muscles only pull, they only contract. Okay. Which seems kind of interesting because people are always like, well, no, you can push or extend muscles. Yeah, but muscles only are forceful to one direction when they contract. Okay, so even when you're bench pressing or pushing, you have muscles that are contracting and forcing your hands forward. Okay, so like the tricep, which you think is going to be an extending muscle, but this is actually contracting as well. It's pulling your hand or your arms out. Okay, your pectoral muscles when you push are going to be shrinking or shortening, causing your shoulders to turn or that humerus to move forward. okay? So muscles only contract, they do not extend, okay? So we talked about the myofibril, the unit, which has dark and light bands. And this is essentially a sarcomere here, one single unit within the muscle. And what you have is a contracted sarcomere here. So you have uh, myosin and actin filaments that contract to make the sarcomere, which we'll see right here. So you have myosin and actin filaments, okay? And what you have is a thick filament, which is myosin, and then actin filaments, which are the thin filament. So myosin is essentially staying put in the middle of that muscle or in the middle of that sarcomere. And the thin filaments are what's going to be moving, right? You're going to have the pulling of the thin filaments or the shortening, okay? So the thin filaments actin is shortened, causing the contraction of the muscle. The thick filaments myosin is going to be that anchor, okay? So think of that anchor, and then the actin is going to be that movement or the contraction of the muscle tissue, okay? Yeah, okay. So in order to contract the muscle, you have ATP binds to a myosin head, right? So here you have the thick filament myosin. And again, these little heads are what's going to stick to the actin filaments. That's going to allow for the contraction to pull. So ATP binds to myosin head, which is then released from an actin filament. So the breakdown of ATP clocks the myosin head. So this essentially, think of it as like a hammer and a gun, or a cocking something, right? Or you're pulling back the bowstring to release an arrow, okay? So this is cocking or pulling back that action potential, right? Or pulling back the hammer and a gun, or pulling back the bow and arrow, okay? So the ATP cocks it, right? Gets it ready, okay? The myosin head attaches to the actin binding site. So boom, you have the release of the actin, and the power stroke slides the actin thin filament toward the center of the sarcomere, okay? As long as ATP is available, the process can be repeated, okay? And the muscle is fully contracted. So in order to get the release and the movement of the myosin and actin filaments, you need to have ATP, okay? And this is important, okay? If you don't have ATP, you don't have the release, you don't have the cocking of the myosin head, okay? One thing to note as well about how muscles work, muscles are stronger when they're contracted, okay? So the muscle on the extension, let's say if you have your arm extended outward, your bicep is at its weakest at its fully extended form. It's at its strongest when it's fully contracted, okay? And same thing with other muscles as well. So when it's elongated, it's not as strong because you have less connection of the myosin and actin, right? You have more connection here, right? You have more surface area, so therefore you have more of these myosin heads binding to actin, okay? So muscles are much stronger when they're shortened already, a little bit shortened. Then they have a little smaller distance to travel, but also there's more connection or more touching of the myosin to the actin filaments, right? Um, so it's important, especially when it's fully contracted. Okay. Um, Another thing to note too is that ATP requires this to be cocked or released as well. So think of what happens when an individual dies. Okay. So we produce ATP through a couple of different ways, either through glycolysis or through cellular respiration, which produces the most ATP, right? Well, if you don't have ATP, and that occurs when someone dies, right? You run out of ATP, you stop metabolic functions in the cells. You also have you also halt the ATP that's available for muscles, right? So what's one of the key things when someone dies? Think of it that way. What happens when they pass away? Let's say an animal. Um, you might come to an animal and it's cold, and one of the first things you'll notice if it's been sitting there for a little while is that it's stiff, right? Even if it's not cold, it's stiff. And people are like, well, that's rigor mortis. Of course, it's stiff, but why? Well, why is the organism stiff after it's dead? Well, think of this. You need ATP to release the myosin head to cock back the myosin filament, right? If you do not have ATP, if you do not have ATP being produced by the body anymore because the organism is dead, you lack the ability to cock the myosin head. So depending on where The myosin head is, depending on if the organism has an extended or flexed muscle, you then release the myosin head because of the ATP not being available anymore. This means this myosin will then release and bind to the actin, and it will stick and it will not release. And when it sticks, it does not release. You have essentially rigor mortis setting in. You have the complete freeze up of those sarcomeres in the muscle tissue, right? And you have the inability to move the tissue back and forth anymore. Whereas if someone were to pass out or someone was just passed away, you have the ability to almost move that body in any direction. And then eventually that ATP gets used up and the muscle tissue sets in. Rigor mortis sets in because you lack the ATP. Okay, so that's important to know, right? So relationship between motor neurons and muscle fibers. A motor unit consists of motor neuron and the one or more muscle fibers that stimulates it. When a motor neuron receives signals from the CSN or CNS, it passes them via synaptic terminals to the muscle fibers, causing the fiber to contract. Right, you have the central nervous system essentially sending that nerve signal out through the peripheral nervous system. That peripheral nervous system has motor units that innervate those muscles at various different synaptic terminals, allowing for the contraction or relaxation of those muscles. Okay, so depending on the stimuli, right? So we have control of this. Okay, we have control of skeletal muscles. All right. Any questions for today? So please make sure you sign up for a presentation. Please make sure that you are getting your papers done. Very, very important. We will be
I do
having.
have a question.
Sure, Nadia, go ahead.
Yeah, I do jog every day. So
Hmm.
should I not do that anymore?
No, no, no. I jog too. I do like cardio or walking or
No, but
I,
like I do feel it in my knees, like it hurts, and I'm like 21. So like, should I just do it like once a week?
um I'm not I'm definitely not someone that can give you I'm not a doctor or anything. I don't know. But
Okay.
that's really, yeah, that's really on you, and you should
Uh-huh.
you should definitely talk to your physician about that. But
Okay.
from my experience, I do have a brother that was he served 10 years in the Marine Corps. So
Mm-hmm.
his knees are shot. Like they are so
No.
bad. So and that came from running, and he had a large pack on his back, and the
Oh,
doctors tell him all the time, your knees are bad. It's probably best that you don't run. But
okay.
you didn't serve 10 years in the military that
Yeah,
you
no.
didn't have a backpack, 100-pound backpack
No.
on your back.
I
So
only carry my phone.
yeah, so yeah, maybe
Okay.
I would talk to your doctor, and maybe,
Okay.
yeah, someone like a running coach too, because that helps running and good technique and stuff. But um,
Ooh, okay. Okay,
yeah,
thank you.
no problem. Yeah, but I'm not, yeah, not medical advice here. I just tell you a couple of quick things about cartilage. I don't know if it's beneficial or not. But anyways, about the topic and the paper presentations and things like that. Uh, Please make sure you sign up. We don't have class Wednesday or Thursday again for the holiday, but I will have a lecture tomorrow on chapter 28, which is, I think, flowering plants. So and that will be the last chapter that's going to be on your exam. Okay, so I will upload some study questions for next week as well for the exam. Um, and I'll probably give you either Monday, Tuesday of next week to complete the exam, or I'll give you like a Sunday, Monday. So I'll send an email out and I'll kind of take a consensus on what we would want for that. Um, Another quick thing: make sure you finish your assignments as well. So you do have a lab that I uploaded. I'm gonna be uploading some questions this week as well for practice. And again, I'm giving you till the 30th to complete everything. Okay, so you have unlimited attempts on every assignment. Um, so you can go back and redo assignments if you want your full points for those. I'm going to kind of leave that up to everyone to see how far they want to take that. So all the assignments are open again. just please make sure you go and you finish the ones that you didn't complete. And you have up until the 30th. So the due date for almost everything is the 30th, if not in December, if it's a new assignment. So just make sure you're getting that done as well. Okay. So again, I've had a couple of students that didn't even know that the presentations were starting today. No one signed up, so there's no presentations today. But please make sure we at least start on the 30th. Okay, of November. People, we have like, other than that, if you don't start on the 30th and you don't sign up, then there's no other days that you're going to be able to sign up for. And you're just going to lose those points, which I don't want for anyone. Okay. That's 20%, right? 20 points. That's a chunk. It's like missing a week of assignments. So please make sure you get that done. Okay. Any questions, please email me. Again, I'll have a lecture again tomorrow on, hopefully we get through the whole chapter 28. Okay. And I'll upload some questions. I'll let you know what the assignment will be on though. I think it might not be on McGraw-Hill. It will probably just be an assignment that I upload on Canvas. So just make sure you get that done as well. Okay. So again, email me if you have any questions.